Here, in the heart of a bustling laboratory, scientists embark on a groundbreaking journey into the realm of regenerative medicine. They are engineering the very building blocks of life itself with a singular purpose: to combat diseases that afflict humanity. At the forefront of this expedition lies the innovative use of genetic circuits to engineer stem cells, guiding them to become specific cell types crucial for treating diseases like cancer and neurodegeneration. Our team has set up cameras throughout the laboratory, capturing the researchers in their natural habitat. The conference room, a location where the scientists present their findings. Let's take a look at what they have been working on recently.
Stem cells are capable of transforming into specialized cells with specific functions. When combined with genetic circuits, they express proteins called transcription factors, which convert them into a different cell type. In this study, we transform induced pluripotent stem cells into immune and neuronal cells.
Our project begins with the process of stem cell culture. This allows cells to proliferate in a controlled environment to maintain their pluripotent state. Doing this also enables us to have a sufficient amount of samples to experiment with.
Next, we designed specific DNA sequences to express genes in the cell using various molecular biology techniques. We combined multiple DNA fragments into a single plasmid containing the genes of interest. When this genetic material is inserted into the stem cells, they will differentiate into macrophages and neurons under the right conditions.
To reprogram our cells, we use genetic circuits to tightly regulate gene expression so that only those that are significant to differentiation were expressed. These gene circuits are inducible, meaning they are controlled by the presence of antibiotics.
To introduce our genetic circuits into stem cells, we use the method called nucleofection. Through an electrical pulse, nucleofection creates pores in the cell membrane, allowing external DNA fragments to enter the cells.
We then tested the integration of the genetic circuit into the cells by using different screening methods to analyze the size and quantity of the genetic material.
To ensure that our genetic circuits had effectively converted the iPSCs to macrophages and neurons, we analyzed the cell's expression with qPCR. qPCR allowed us to obtain real-time data that proved that certain genes associated with our desired cell types were turned on in the cell. This research has the potential to increase the effectiveness of certain immunotherapies, as well as contribute to the creation of novel personalized immunotherapies. For example, these therapies can utilize gene circuits to reprogram cells to recognize and destroy cancer cells, offering a promising approach to cancer treatment.
By creating our own neurons and macrophages in the lab, we can model diseases such as Alzheimer's and test the functionality of our genetic circuits in the cells. By confirming that our neurons are able to grow and survive with the engineered macrophages, it poses a potential therapy for neurodegenerative diseases.
To efficiently reprogram iPSCs into macrophages and neurons, the right transcription factors must be identified to express in the cells. Because of the large variety of transcription factors, we will develop a machine learning algorithm to select only the crucial proteins required for our genetic circuits.
Throughout their time in the laboratory, our camera crew's relentless efforts to capture the scientists without being caught was impressive. We were able to bring to you the authentic livelihood of this species inhabiting the bioengineering floor at NYU. And so in these moments of discovery and collaboration, we witness the relentless pursuit of knowledge, driving us ever closer to unlocking the mysteries of life itself.